how safe are stem cells that replace beta cells. This was during the discussion downstairs.
Yeah.
And in diseases where you can do ethically more, like in Parkinson's, that's where we're going to learn whether these stem cells are going to grow some things untowards or unwanted, and that's going to open the door for these type of stem cells.
You're talking about the potential for transplanting iPS cells turned into dopamine-producing neurons into the brains of Parkinson's patients.
Exactly.
And so that would actually give you information that would be
That's
pertinent.
Right.
Okay.
For heart attack and stem cells when they regenerate, myocardium and these type of things. It's an exciting field. If you would have asked me whether all this is possible, you know, I would have thought, you got to be kidding. We cannot differentiate all these stem cells in these different tissues, and there we go. So research is quite exciting in many ways.
So this is still fairly recent. I mean, it was what in 2007 that iPS cells were discovered.
I know. Isn't this cool how that has moved? It's an exciting field. It is quite the field. But you're right. I mean, what we discussed before, these cells in beta cells, their body is still sensitized, so we still need to deal with the immune system. And part of the research we're doing here is to basically understand, and I gave you some papers on that, what constituents of the immune system are present in human pancreata as the hallmark of diabetogenesis in humans. And this has only recently become possible since we are part of a big cooperative consortium that's called NPOD, and it's a donor for pancreatic organ donations from humans that get collected to transplant OPOs. So if somebody is an organ donor, has type 1, so the pancreas cannot be given to a recipient because beta cells not there or destroyed, and this pancreas will go to Gainesville. The other organs will still be donated. And these pancreata we can do much better research on than pancreata that are collected post-mortem and then are not as well preserved. And we learn a lot about the disease this way. It's actually quite phenomenal. And in this way, the ideas, of course, the more we learn on the basic side, the better we can work on the afferent side to say how drugs should be targeted. So let's say you want to help a adjuvant for these peacekeeper cells to block a certain cytokine. Well, that cytokine is not even in the pancreas. That's probably not the one you want to block. You want to block one that's there.
Yeah.
But if you don't know who's there, you don't know what to block, and it's like more like a shot in the dark. But
so you got to get those addresses.
You got to get the addresses. Once you have the addresses, you can do it in the target fashion. And I see this as a reiterative process. The coolest thing is that Novo was one of the few companies who actually realized that making new drugs for type 1 is not a process where you have to find the latest and greatest and most secret.
That's Novo Nordisk, right?
Yeah.
Okay, yes.
But you have to work together. They have this really embraced this concept, and they also embraced the concept of understanding the pathology of type 1. And that's why they let me keep this work here at La Jolla Institute, because they said, well, everybody will learn from that, not only us, and it's something that the public should know. And it will not be one single person or company who will learn the pathology. The whole world needs to do that. And it's a scarce resource. 140 pancreata pre-diabetic worldwide since 1925. That's it. That's our organ access. 125 or so. Right? So you can imagine, if you ask, why don't we understand the disease? It's not as simple as having an arthritic joint. You take a little thing out of the thing, you do some biopsy, you learn what's going on there, and you can do hundreds, and you can do your own study. 125 pre-diabetic pancreatitis.
How long can you keep those alive for once you get them? You take them from a corpse and
no, you have to. They are preserved, frozen, and then it has to be all histology. But if they are frozen immediately, at least you can do RNA studies and all these good things. You can imagine if the pancreas is harvested and it's in a person who died under certain circumstances, and it takes some days before the body is released, maybe. Yeah. If it sits there for days, then it's changed. Everything changes. The composition, there's autolysis, there's a digestive organ. You can't study it anymore, right?
Yeah. There has been talk about creating a bioprinting organs, not only just for transplantation, but for research. Any progress in creating a living, sustainable pancreas for SETI purposes?
The only thing that we have is the cells from ViaCyte. I think a living, sustaining organ for study purposes is going to be hard because it's a digestive organ. As soon as you annoy the pancreas, it makes a big inflammation because you get these digestive juices leaking. It's like if you get pancreatitis, you know it's a severe disease in humans. Just because of the digestive juice in leaking in the retroperitoneal, it's a big mess, right?
There was also some studies about that some of the deaths from shock are caused by autodigestion when these juices get into your bloodstream and start getting into all these organs.
That's right. That's right.
Okay. All right. Any final thoughts? Anything else?
No. Just thank you that you came and you're very knowledgeable. I like this.